Good afternoon, everybody. It's 1 o'clock, so I'm going to try and keep us on time. My name's Graham Lipkin. I'm a kidney doctor looking after an adult cystinosis clinic in Birmingham in the UK, and I'm going to be the chair for this session. This session is one of the highlight sessions of the meeting. I mentioned this morning that we have had a fabulous scientific session yesterday of clinicians and of scientists. And so we're going to give you the highlights of that meeting today, things that have a particular immediate impact on patient care. So the organization of the session is we'll have two talks. The first two are focused on both clinical and experimental aspects of the prodrug CF10, after which we'll have a short question and answer session. And then we'll move on to 4 more talks after that, and then have another question and answer. So after the first two, there are roving mics. Please feel free to ask questions. You're very welcome, and we'll try and answer them for you. So it's a real great pleasure to introduce our first speaker, Professor Herbie Newell. Many of you will know Herbie. Herbie is the Emeritus Professor of Cancer Studies at the University of Newcastle and the Professor of Drug Discovery at University of Sunderland. And Herbie has very effectively and dynamically taken on the mantle for the development of CF10 after the very sad loss of Rosalind Anderson. So, Herbie, thank you very much.
Thank you very much indeed, Graham, and thank you all for the invitation to come and speak to you today. Earlier on, we were asked about one word that you would use to describe this meeting and this community, and the word that came straight to my mind was inspirational. You really are, cystinosis patients and your families and carers, a real inspiration to all of us involved in biomedical research. And for me personally, it's been a phenomenal honor to be able to inherit, as Graham said, under tragic circumstances, the CF10 project from Ros Anderson, and to realize this project not only as a much better therapy for cystinosis patients, but also a lasting memorial to a truly great lady. So I'm going to give you an update on where we are with the development of the cysteamine prodrug CF10. And Eleanor very kindly earlier on gave you a brief insight into this, but I'll give you a bit more detail. So you will be aware, painfully aware, of the drug cysteamine. It's a very useful drug. It's actually, of course, the only proven effective treatment for cystinosis at the moment. It works by removing cystine. It's a very simple and relatively cheap oral medication, and it has actually a very elegant mechanism of action, as we heard earlier on this morning. But, and of course there is a massive but, it's an absolutely terrible drug to have to take in terms of the side effects, which are listed here, and I won't reiterate them because you live with those side effects day in, day out. Probably the most important concept in pharmacology, and definitely in drug development, is the concept of therapeutic index. It's the ratio between an active dose and a toxic dose. And for a good drug, that ratio should be big. So the dose you need to take to treat whatever problem you have is much, much lower than the dose which would cause a side effect. And so if you've got a headache, and I was a very good boy last night, and the generosity of Cystinosis Foundation UK at the dinner provided us with excellent food and drink, and I moderated on the drink, so I didn't need to take any paracetamol this morning. But if I had taken a couple of paracetamol, it would have worked, and I would know that for that paracetamol dose to have been dangerous, I would have had to have taken an awful lot more than the 2 tablets that would control the headache. And so it's got a very big therapeutic index. I know through talking to patients, carers, and parents here that essentially no patient taking cysteamine has no toxicity. There are always side effects. And so the best thing we can do with cysteamine is consign it to history, and that's what we're trying to do. And the late Ros Anderson came up with a really elegant way of approaching this problem, and that's to design what we call a prodrug. And a prodrug is a chemical which is inactive in its own right, but after you've taken it and it's gone into the body, it's converted to the active species. And this is the prodrug that Ros developed. So it has, in red, the active drug, cysteamine. It's attached to a glutamate group, which targets that cysteamine to the tissues, and that's shown in blue. And then 2 other modifications are needed. First of all, there is an ester group shown in light blue, and that's to cover up the SH sulfhydryl group in cysteamine, which, because it is exposed in cysteamine, gives the problem of irritation, the problem of ulceration, which indeed can sometimes progress through to perforation, actually holes in the gastrointestinal tract. But by covering up that with an acetyl group, you prevent that problem. And lastly, it has an ether group. That was the green thioester group. Lastly, it has an ester group, and that is to make sure it's absorbed really effectively from the gut. The key thing with a prodrug is it handled in the body the way it was designed to? And so the first series of experiments we did are what we call metabolism and pharmacokinetics. This is looking inside the body after you've given a drug to see what the parent drug, CF10, has been converted to. And in our case, the critical conversions are, first of all, to remove the thioester and the ester groups, and then at the tissues to remove the glutamate group. And the first series of experiments, which Ros did before she died, was to look at this metabolic process in vitro. So that's in a test tube in the lab. This is before you've got as far as doing any animal studies. And so what Ros showed was that there are 2 enzymes we call esterases that very effectively clipped off the ethyl group and clipped off the thioacetyl group. And then there's another enzyme called gamma GT that is present throughout our bodies on every cell that clipped off the glutamate group. And that enzyme is on the surface of the cell. So the cysteamine would be carried in the blood and extracellular fluid to cells throughout the body, and then it would be cleaved off by loss of the glutamate group, and you would deliver that to the tissues where the cysteamine is needed. So that was the theory, but is there evidence preclinically that that's the case? And this first graph here shows the levels of cysteamine and this critical intermediate, gamma-glutamyl cysteamine, in the blood of rats given CF10 orally. And the line at the top is the intermediate gamma-glutamyl cysteamine at very high levels, and down the bottom are the levels of free cysteamine in the blood. And what we wanted were high levels of the intermediate and low levels of free cysteamine. And that's because by having low levels of free cysteamine, you prevent the cysteamine having all the nasty side effects that you get when you take cysteamine directly. And that's, of course, here, as you can see, exactly what we saw. So this is great. It was behaving metabolically as designed, but critically, does it actually deliver cysteamine to tissues? And so we use the kidney, obviously a very important organ in cystinosis, to answer that question. And this shows in the top line is the concentration of cysteamine in the kidney, and the bottom line is the concentration of cysteamine in the blood. And then here again, you can see is a very big difference. And I should have pointed it out on the previous slide that the y-axis going upwards is a log scale. So there's a massive difference between the cysteamine in the tissue where you need it and the cysteamine in the blood where you don't want it. And that, again, as I say, is exactly as Ros designed this molecule to behave. So that's great. It's handled metabolically okay, exactly as designed, but critically, does it still work? And before she died, through many collaborations with this community, Ros generated some cell line data and very limited data in a mouse model and some zebrafish data. But recently, as you've heard already, some excellent new models have become available. And we've had a fantastic collaboration with Jen Hollywood, originally while she was in Auckland, and now that she's back in Ireland in Cork, looking at CF10 in her rat model. And this is how that model is generated. It's so-called gene editing. And it's produced a model of the disease which mirrors extremely closely the clinical progression of cysteamine and the clinical of cystinosis and the clinical features of cystinosis. And so using that model, just in summary, looking at all of the characteristics of cysteamine, which we know well, we've asked, is CF10 as good as cysteamine? And the answer is yes in every case. So in terms of ability to deplete cystine, in terms of ability to prevent excessive water consumption, prevent excessive urine production, the incidence of Fanconi syndrome, the histopathological and pathological effects on the kidney, and then the overall effect on weight gain in these animals, the two drugs, when they're given at the same equivalent dose, which we call the molar equivalent dose, are equiactive. And as indicated in the bottom text there, because CF10, as I'll mention in a moment, is so much less toxic than cysteamine, we're optimistic that by increasing the dose, we may be able to get even more activity. And that's something that we're looking at the moment. So this is, apart from these most recent in vivo data, where Ros got the project to at the time she was unfortunately diagnosed with a very aggressive uterine sarcoma. So as I took the project over, the first thing that we did was to apply to the Medical Research Council for funding to undertake the regulatory preclinical safety studies that you have to do before you can take a compound into clinical trials nowadays. And Ros was able to apply for that money in her name before she died. And the week before she died, she received a letter from the Medical Research Council, and I'll choke here because it always upsets me so much, that her project had been funded in full to the tune of £1.6 million, and it was the highest ranking project they had seen in the last 5 years. And so Ros, bless her, left us knowing that the project was going to go forward. And with that funding, we were able to undertake a very comprehensive series of studies. It's a legal requirement in most jurisdictions that you have safety data in a rodent and a non-rodent species. And industry standard is the dog and the rat. And so we followed the required procedures. Had we not done this, we would not have been able to subsequently make CF10 available in all jurisdictions around the world. I appreciate people have very strong views about the use of animals in biomedical research, but this was a go, no-go, non-negotiable point. So these studies are all undertaken under the strictest animal welfare conditions, and they're an unfortunate necessary step in the drug development pathway. And the results were nothing short of spectacular. So in the dog, where the drug is given as a capsule formulation, which will be the initial clinical way of giving CF10, we didn't see anything at all at doses up to and including 200 milligrams per kilogram for 28 days. If we went above that, we did see some minor signs of gastrointestinal toxicity, nausea and vomiting. But these are truly industrial doses. Critically, and this is a really important point and a result I was so pleased to see, if you look at the half-life of this key intermediate, gamma-glutamyl cysteamine, in the blood that is carrying the cysteamine to the tissues all around the body, the half-life is 4 hours. That's really important because it means that you won't have to take the drug more often than twice a day. And when we get to humans, you might predict the half-life will be a little bit longer still. So there's a very good chance this will be a once-a-day medicine. We did studies in the rat as well. The no adverse effect level was 333 milligrams per kilogram. This is at least 4 times higher than the dose that was as active as cysteamine in the rat model that I showed you the data for at the moment, a few moments ago. So we have a drug here that is as active as cysteamine, but has, we would predict, absolutely no toxicity, and no adverse effect means no adverse effect. So specifically, there will not be any hematological toxicity, no effect on your blood counts. There'll be no effects on clinical biochemistry, so no effect on liver function, for example. And we could detect nothing pathologically when tissues were removed and looked at under a microscope. This will, of course, be a lifelong therapy, and so we need to prove that there is no risk of genetic damage because that could lead to cancer. Now, there's no reason to believe this would be the case because cysteamine does not produce a significant increase in tumour rates, we know, and the other components all of us in this room take all the time. There's glutamate. Well, we all take monosodium glutamate every time we have some soya sauce on our Chinese meals. There's an acetyl group. Well, that's vinegar. If you've ever had fish and chips, you'll have had some of that. And then there's an ethyl group, which is coming, of course, from ethanol. And last night, I freely confessed already, I did consume some ethanol. So there's no reason to believe that there's going to be any toxicology in that respect. And then critically, you also need to look at the heart and look at respiration. Again, completely clear. So we were really, really reassured that there would not be a problem with the compound. Now, we're very aware, of course, that a major issue for cystinosis patients taking cysteamine is the problem of the unpleasant body odour and halitosis, which brilliant studies by colleagues in Leuven demonstrated are due to 2 metabolites of cysteamine. And so particularly methane thiol and dimethyl sulfide are produced in the body from cysteamine after you've taken cysteamine. And so we wanted to know whether or not CF10 could produce these metabolites, because we would hope that by keeping the blood levels of cysteamine very low, we wouldn't see the production of these metabolites at significant levels. The data on the next slide are a bit complicated, but that just reflects the challenges of measuring the levels of these volatile gases in the breath of dogs. So in our dog toxicology study, we looked at a group of dogs who were receiving the no adverse effect level, so a dose that was completely non-toxic, 200 milligrams per kilogram per day for 28 days, and compared that to a control group who were not receiving CF10. And we looked at the animals on minus day 7, so that was before they'd have any cysteamine in this group here. On day 1, before they had any CF10, not cysteamine, and then after 28 days dosing. Again, this is the control group, and this is the treated group. And these are 6 animals, and what we're looking at is the range of the levels that we detect. And you will see in brackets, if we look at the control, let's look at these controls on day 1. These animals have had no CF10 and won't ever get any. Most of the animals actually had no detectable levels of this very smelly metabolite. But one animal had a very high level. Where did that come from? Well, any of us who have ever kissed a dog will know that kissing dogs can be a little unpleasant. Dogs have bad breath occasionally. And so that's what this is. It's just the normal gut bacteria of the dog that by digestion of or metabolism of the food is generating some noxious compounds. However, critically, if we look at this group here, there is absolutely no difference in the exhalation of dimethyl sulfide after 28 days dosing at a sizable dose in comparison to the untreated animals or the same group before treatment. So the same data are true for the other metabolites. So we're pretty confident that there is not going to be a body odor and halitosis problem with our drug. So you've seen this already. This is where we are. I was really pleased to hear from Professor Emma yesterday that at this meeting in 2006, Ros presented the design of CF10. And I know it's 18 years later, but it's great that it's here. We're talking about it transitioning into the clinic. And so Ros described the design and then the discovery of CF10, and we're now right on the cusp of going into a clinical study. So what we're doing, firstly, we're doing some more studies in Jen's excellent rat model with higher doses to see what that shows. And then we're going to undertake a single low-dose CF10 pharmacokinetic study in cystinosis patient volunteers. And the reason that we're going to do that is exactly the same reason as I gave when we did our first rat study, and that is to prove that the drug is handled by your bodies in the same way as it's been handled by the rats and the dogs in our preclinical studies. Because if that is not the case, then we need to go back to the drawing board. Now, I have no reason to believe that it won't be the case, but we do need to prove that. And that has a number of significant additional reasons. It's not only good science to do it that way, but ethically it's really important because, as we'll mention in a minute, the initial phase 1 and phase 2 subsequently clinical trials will require a major commitment from you as cystinosis patients to take part in them. And we don't want to ask you to make that commitment unless we're really confident that the drug is working in the way it's designed to work. There's also an issue of logistics. A big phase 1 program in probably multi-centers as this will be is going to take some serious organising. There's also going to be a financial driver for doing this because the phase 1 program is probably going to cost best part of £5 million, whereas this initial study is going to cost about £250,000. If we relieve £5 million from the funding agencies, it means that that money is not available for other equally important areas of biomedical research. So we want to be really confident when we ask for that money. And not coincidentally, but importantly, having these data will really increase the likelihood that we'll get that money. So this study will, as I say, be 3 cystinosis patient volunteers, a single dose of up to 3 100 milligram capsules, blood sampling over 6 hours, analysis of the blood samples for these 2 critical components. And we are now at the end of the manufacturing of the material, which has to be to a special quality standard called Good Manufacturing Practice, or GMP. We're just about to start making that drug into capsules, and then the patient treatment and the sampling will be possible. We hope that we'll be all ready to go by the end of the year and treat the first patients at the beginning of next year. There's a lot of stages in making this happen. The funding has to come from somewhere, and I'd like really to acknowledge Sunderland University here, who have put their hands in their pockets and provided £250,000 to make this happen quickly. We could have gone off and written grant applications, which would have gone to and fro, lots of questions to answer, all of which would have taken time. But Sunderland, to my mind, very admirably took the decision, let's cut to the quick, here's £250,000, go for it. The synthesis, as I say, is well underway, nearly finished. Manufacture's about to start. There is, as we heard in the earlier discussion, a massive amount of paperwork to be gone through to make all of this happen. Legal agreements, clinical protocols, lab manuals. We have to find someone to sponsor the study, take ultimate responsibility. And I'm really pleased to say it's ticked to all of those. And then we need ethics committee approval. And that's the last thing that we need to do. And then the patient treatment and sample collection can start. So what does it actually look like when you make a drug for administration to patients for the first time? So on this slide there is a photo of colleagues at the High Force Research in Durham who are making it. In case you're wondering why they're all dressed up in what look like hazmat suits, that's not because the drug is dangerous and they're protecting themselves. It's because they are protecting the drug from themselves. We're all covered with bugs and other things that you probably don't want to look at under a microscope. They can fall into a drug while you're making it really quite easily if you don't cover yourself up properly. So that's what that's all about. And the formulation will be done at RVI Specials in Newcastle. The treatment of the 3 patient volunteers will be done by Graham at Birmingham, and then the analysis in a contract research lab. And so we are very, very nearly there to start that. In terms of a schematic, what that looks like is patients who volunteer to take part will be consented, will carry on taking their usual cysteamine dose, and then 2 days, 48 hours before the CF10, they'll stop taking the cysteamine to allow it to wash out of their system completely. Then we'll give the CF10, take the blood samples for 6 hours, and patients will go straight back onto their normal CF10 dose. And we are confident that that brief window without cysteamine, 48 hours, will have no significant impact on the management of one's condition. So please rest assured about that. The next steps after this will be a pilot run-in where we're going to do all of the laboratory investigations that will be part of the phase 1 trial of CF10, but in patients who are taking cysteamine. So this is just to sort out all of the logistics, because when we get to the phase 1 trial of CF10, we don't want to be developing and validating assays. We want to know that everything is going to work smoothly. So again, we will be asking for patient volunteers for this study, and we've had really great interaction with the patient community, the cystinosis community, in putting this protocol together, which again, the study will be done in Birmingham. And then once we know that everything logistically is going to work, we'll be ready to start the phase 1 trial, which we're anticipating will be, we hope, at the beginning of 2026. We've already sent an outline application to the Medical Research Council for funding. They loved it. £4.6 million, they're really keen to get in our back pockets. We now just have to put this final PK data from the pilot study in and submit the full application. And what this study will look like, there'll be 2 components. The first component will be 7 days of treatment. And again, we will consent patients with cystinosis to take part. It's really important to emphasize the reason this is being done in cystinosis patients and not healthy volunteers is measurement of cystine levels is absolutely critical to knowing whether or not we've got the right dose of our prodrug CF10. And you can't measure cystine levels in healthy volunteers because there aren't raised levels, of course, in healthy volunteers. So patients will stop taking their normal cysteamine. They'll then not be treated for 7 days to allow the cystine level to rise a little bit. So we'll then treat for 7 days with CF10, and we will gradually increase the dose of CF10 until we start to see the cystine levels come down. So we know we've got an effective dose. After 7 days, patients will return to their normal cysteamine dose. And then when we know we've got a dose that works, i.e. depletes cystine, we'll then swap to 28 days dosing, because once you have got 28-day exposure, you're then able to move on to continuous dosing, which is, of course, ultimately how we'll use CF10. And again, we may look at the dose level here to make sure that we're giving a really effective, non-toxic, completely safe dose. So what's this going to mean for you as cystinosis patients and for cystinosis patient carers and parents? Well, we hope this will be a palatable and easy to take medicine, that there will be reduced side effects, specifically damage to the stomach and the gut, and the problems of bad breath, halitosis, and body odor will be resolved. We should have improved tissue cysteamine delivery and hence a lower chance of kidney failure and extrarenal effects, with a reduced need for dialysis and transplant. Fewer doses should be required because of this increased half-life that we're seeing. And overall, there should be an improved quality of life with knock-on effects of reduced psychosocial issues. And then lastly, overall, this will be a better treatment because there will be a reduced risk of failure because it will be taken more consistently. And all of this will be delivered due to the foresight of the wonderful Ros Anderson. In conclusion, just to note the current project team working on this, I'd like right up front to really thank Cystinosis Foundation UK, not just for their continued support, but for the fantastic foresight that they had in supporting Ros over many years to the tune of that £1 million to take CF10 from a concept to a clinical entity. And for a rare disease that the pharmaceutical industry would never invest that type of money in for drug discovery, it's, I think, an outstanding testament to the commitment of Cystinosis Foundation UK to generating a better medicine for this condition. So thank you, Cystinosis Foundation UK. We've then got the team at Sunderland University that I work with, the team that we are now working closely with at Birmingham under Graham to actually take this into patients for the first time. And then there's a huge number, an army of people out there working in contract chemistry companies, making CF10, in safety and toxicology studies, doing all this animal testing. The colleagues at RVI Specials at Newcastle, and then colleagues at Sundara Pharmaceuticals, who will ultimately, when this becomes a licensed drug, be responsible for marketing it. I estimate that at the moment, working on CF10 to get it to you, there are probably about 100 people in these different organisations. And so I'm told that we may have somewhere in the region of 220 cystinosis patients in England. And so probably half that number are beavering away in labs, bringing this new treatment to you. So I really hope that you feel that there is hope coming in the future. And then for people who outside the Sunderland current collaboration, people who worked with Ros previously, many of whom, of course, are in the room in various different areas. And again, I'd really like to highlight the work that we've done and are doing with Jen Hollywood on her brilliant rat model. And then last but not least, the funding bodies. So on taking this on, I was, of course, very pleased to help Ros at a terrible time in her life. And that, I will tell you, I'll be honest, has been a little bit of a responsibility on occasions. But I only need to come to a meeting like this for half an hour to realise it's worth it 1,000,000 times. So thanks for listening to that. And we are nearly there. Next time, we hope we'll have clinical data to show you. Thanks very much.
Fabulous talk, Herbie. You are the man, and we wish you Godspeed in further development of this. Before we take questions, I'm going to invite the next speaker up because the two talks link together. You've already heard a little bit about Dr. Jennifer Hollywood. Jen is got a PhD at the University College in Cork, and then left us for a decade to go to Professor Alan Davison's lab in Auckland. Thankfully, she's now returned to this side of the world, and she has been involved in developing animal models and using those animal models for future drug development. So it's a real pleasure to invite Jenny to come up and talk, and we'll then have question and answer session with Herbie and Jenny. Thank you.
Thanks. Oh yeah, it's perfect. So yeah, thanks very much for the organisers for inviting me here today. It's really good to be here in person because the last couple of times I had to do online Zooms and things like that. So it's really nice to be here. So I'm going to talk today about some preclinical studies we're doing in the rat model that Herbie already mentioned. And as you know from Herbie's talk, cysteamine, and I'm sure you all are experts in this, cysteamine, although it does have advantages of depleting cystine, it does have many disadvantages. So one of the big problems is addressing these side effects. And thankfully, I think Herbie and the team have nailed that side of things. But another aspect of cystinosis that we need to address is the fact that cystine depletion is not the only thing we need to look at. Because we know that taking cysteamine, even from birth, the patient will still progress to kidney complications and other complications. So there's something else going on that we need to look at and treat. So what we were doing in Auckland was modelling cystinosis using induced pluripotent stem cells. And these cells are like embryonic stem cells in terms of the fact that they have the ability to become any cell type of the body. However, they are derived from an adult person. So we take a skin cell biopsy, and then we reprogram these cells by culturing them in a dish and adding 4 genes. And then these cells reprogram back into a stem cell. And what happens then is you can coax these cells to become any cell type you want. So you can make heart tissue, lung tissue, but what we're interested in obviously is kidney. So we took these cells, we added a couple of factors, and they transitioned into what we call embryoid bodies, and then they further transitioned into these little kidney organoids. And these are mini kidneys in a dish, so they have all the cell types of a kidney that you'd need, such as the glomerulus, the nephrons, and the other proximal tubules. And this is a really nice model then that we can study, and it's human tissue, so it's relevant. So what we did was we took these stem cells and we used a gene editing tool called CRISPR, and we knocked out the cystinosin gene to make a cystinotic stem cell model. And then we coaxed these cells to become kidney organoids. So we had these kidney tissues. And by looking at these stem cells and kidney organoids, we found that as well as increasing cystine levels, having lots of cell death and large lysosomes, they also had this defect in a pathway that's very important for a cell. And Stephen spoke about this earlier, but I'll just go through it again. So in your cell, which you can say is your house, you generate rubbish, and you need to get rid of that rubbish. So you have these little garbage trucks, and this is what an autophagosome is. So it goes into the cell, or it's in the cell, and it sucks up all the proteins and the old parts of the cell that it doesn't want anymore. And then it transports them to the recycling center. And the recycling center is the lysosome. So the lysosome then will break down the components in the garbage truck, and then it will recycle what's useful to the cell back into the cytoplasm to be reused. And what we found was there was as well as having cystine accumulation inside the lysosome, they also had a defect in the autophagy pathway. So they were unable to fully recycle these components. And this block was causing the cell to become sick and other host problems. So we looked at being able to induce this pathway. And one way to do that is by inhibiting mTOR, which you heard about earlier as well. And to do that, we used this drug called everolimus. Now, everolimus is already a drug used in immune suppression and also in treating different types of cancers. So it's an FDA-approved drug. And that was one of the reasons we chose it, so we could repurpose it. And when we combined this with cysteamine, we found that we could reduce or rescue all the cystinotic phenotypes or the symptoms in these cell models and the organoid model. So this was really good proof of concept, showing that in a human tissue, we're able to restore the the way of the cell back into a healthy cell type. So we wanted then to test this treatment in a relevant animal model. So we produced our own rat model of cystinosis, again using gene editing techniques, which Herbie talked about. And this rat model turned out to be really good in that it modeled the human progression of disease very well. So you can see here in this graph, this is cystine levels, and this is the age of the rat. So at 3 months, we started our measurements, and it went from 3 to 12 months. And you can see in white, the white bars here are the cystinotic rats, and the black is the wild type. And they had markedly more cystine accumulating in their kidneys. And this is a picture of a lysosome under electron microscope, and this is the crystal when the cystine forms crystals. That's what it looks like. And then we measured things for Fanconi syndrome, such as urine output. So again, in gray here is the cystinosis animals, and in black is the wild type. And you can see that from 3 months onwards, they were progressively urinating more compared to the wild type littermates. And we measured things such as total protein, glucose, and calcium in these rats. And again, we saw that these were all increasing as time went by, showing that they were losing these nutrients in their urine. And then at dissection, we saw that the kidney itself was markedly different in the cystinotic animals compared to wild type. So we did this preclinical study in Auckland for the past number of years, and I'll just talk you through the study design. So these are the rats here. They're Sprague-Dally background rats, so they're white with red eyes. We gave them the drugs in these little jelly pills because usually when you give a drug to an animal, you have to give it either force feed it or give it in drinking water, which isn't very accurate. So we devised this jelly pill system so that it was a lot more palatable for the rat. And because we were giving them daily doses, it's a lot nicer than giving them oral gavage. So cysteamine, we chose this dose. It's a much lower dose than what patients would receive. And that reason for that was because we wanted to see any additive effects of the second drug. We didn't want it to be totally wiped out by cysteamine. And we gave this twice daily. So every morning at 6 a.m. and 6 p.m., these rats received a jelly pill of cysteamine. And then another cohort of rats received everolimus. So they received 10 milligrams per kg twice weekly. So every Monday and every Wednesday, they got an extra jelly or one jelly everolimus. And then the next cohort of rats received both treatments. So they had cysteamine morning and night, and then every Wednesday or Monday and Wednesday, they got an extra jelly of everolimus. And then we had our control group, and they just got vehicle jelly. So that was just jelly with no drugs. So these guys were getting this drug for 6 months. So we started at 6 weeks of age, and we went to 6 months. And at monthly time points, we collected urine, blood, and we took their body weights weekly. And then at the end, we did these analyses to see if there was any improvements. So first off, I'd like to show cystine depletion. So in black here is vehicle, cysteamine on its own, everolimus on its own in blue, and then the combination in purple. And just this is showing here the amount of cystine in the kidney tissue. And you can see compared to the animals that received no drugs, cysteamine reduced it by about 50%, but the combination of everolimus and cysteamine together had an additive effect of bringing the cystine much lower again. We looked at other tissues such as the heart, liver, and lung, spleen, brain, and muscle. And you can see here the heart, liver, spleen, and brain all had better reaction or better depletion of cystine from their combination treatment instead of cysteamine on its own. And in the muscle then, we did see an increase, but there was a lot of variability in these samples, and we're actually re-looking at those now. In terms of urine output and water intake, again, here is the vehicle group in wine. Blue is everolimus and cysteamine in red. And then the combination you can see here is much lower than the vehicle and everolimus groups and lower again than cysteamine. And again, for urine output, there's again a nice reduction in the output of urine following this 6 months treatment. We looked at Fanconi syndrome markers such as total protein, and we saw again that the combination treatment significantly reduced the amount of protein in the urine compared to cysteamine on its own, and also glucose and calcium. And just when you look at the trend of this increase, so in here, this is on no treatment, and you can see there's a steady increase trajectory of the Fanconi syndrome over the life course of the rats. whereas with our combination treatment, this is much more reduced and leveling out. When we looked at the histology of the kidneys, we can see here compared to the wild type, again, the vehicle, a large and pale kidney. Cysteamine was able to bring back colour. Everolimus didn't do much on its own, but the combination brought it back to a much more wild type looking kidney. And this is just the weights of the kidneys. And then we looked at a histological level. So this is looking at when you do slices of the kidney and look at it under the microscope. And you can see here, this is wild type, so you get this nice histology of the kidney. So this is the glomerulus here, and these are proximal tubules, and they're meant to be nice and full and not much white gaps. You can see here in the vehicle animals, the cystinotic animals, a lot of this white space, that's dilated tubule, a lot of degeneration and loss of cells. Cysteamine was able to reduce this amount of white space, as was everolimus a little bit. However, the combination therapy, you can see compared to the mono treatments, had a better effect on the histology. And then we did this histological scoring. So this is just taking 10 pictures of each section, and then we do these counts, and we have this criteria. So we're looking at this proximal tubule atrophy, which is this disintegration of the proximal tubule, and then dilation and effacement, just losing these cells. And we give this score. So depending on the number of tubules that are affected, they get a percentage score, and then you get a color code. So red's obviously really bad, orange is moderate, mild, and then green is none. And as you can see here, compared to vehicle, which is untreated animals at 6 months, with the combination, we were getting much better reduction in basement membrane thickening, tubule dilation, and effacement, and a better effect on proximal tubule atrophy compared to the mono treatments. And again, I'd like to reiterate that this dose was low compared to what you could give in clinical setting. So to summarize, the combination treatment versus mono treatments, in all our parameters of urine output, water intake, Fanconi syndrome markers, cystine depletion, and kidney preservation, they all did better with combination treatment. I just want to note that there were some unwanted side effects of everolimus, which are known in the field. So everolimus can cause weight loss, which it did in our rats, and we've since looked at a lower dose of everolimus and repeated this study and shown that it can be mitigated, so the weight loss has gone away, but we still get the same positive effects on our parameters here. To conclude, I'd just like to say the combination treatment again, compared to everolimus and cysteamine, over 6 months treatment showed a better reduction in cystine levels, enhanced improvement in Fanconi syndrome markers, and kidney appearance was better compared to treatments alone. And we're also completing studies with the combination of CF10 and everolimus, which are ongoing. We're going to start some new ones in Cork, but in Auckland, we're doing these studies, and the first round of results will be in I think, September. And then I think that these new treatments have the potential to improve lives of cystinosis patients. And I really hope that one day we can get to the clinical stage. And just like to thank the lab in Auckland. So Professor Ali Davidson, who was my supervisor, Pang Yok Chung, who was a PhD student, Jawa, who's our technician, and Robert now has also joined the team, and also our collaborators and our funders, of course. And yeah, thanks very much.
I'm going to invite Herbie back as well. And we'll, so if Herbie joins Jen. So we'll open the session up now to any questions. We've got some roving mics available. Please feel free. There's 2 hands up over here at the back and 1 in the middle. So if you're happy to, just say who you are and where you're from, and we look forward to your question.
Hi, I'm Lauren, and I'm from the UK. I have cystinosis. My question is, the first clinical trial for the prodrug in Birmingham, is there a reason why they're all male?
Yeah, it's an extremely good question. So we want to do this as quickly as possible. And Whenever you treat a woman of childbearing age, there is always the possibility, and I appreciate in cystinosis this is a separate and important topic, but there is always possibility that there could be a pregnancy that is undiagnosed or not diagnosed, unknown about at that time. And so it's really just to do this as quickly as possible. And a similar answer around children. Why aren't these first 3 patients potentially being a child? Exactly the same explanation. With a child, understandably and correctly, there are a number of additional hurdles you have to get over before you can offer a child an experimental medicine. So we're going for the quickest and simplest way of getting to this critical proof of principle answer that will allow us then to get into studies which absolutely will be both male and female to start with, adults to start with. But the moment we're confident things are looking good, we'll write that paediatric protocol and get going on that as well. Thank you.
Hi, I'm Avril from the UK. My question was very similar, but just regarding that, how long do you anticipate the male trials to last, and when would you be looking? Would you be looking at like further volunteers to be involved in the trials after that? How would one get involved?
Another brilliant question. So because it's a single dose, we hope we'll have the answer to this initial 3-patient study by Easter next year, which will allow us to get in the application into the MRC. We're going to make enough drug in this run-in 3-patient study to start the phase 1. So we're hoping that early 2026 we'll be in a position, if we can get all the paperwork done and all the permissions in place, to actually start the phase 1 trial, which will be both men and women.
Thank you, Herbie.
So, guys, thank you so much for just incredible presentations. I heard both of these yesterday and kind of formulated some questions that were in my head, but it seems like both of you are kind of attacking the same question, like how can we reduce the toxicity of cysteamine, reduce the side effects of cysteamine. And so there's a lot of through lines to both of these pieces of research. I'm wondering, is there any benefit or have we thought about actually, as CF10, right, moves forward, is there any benefit to looking at everolimus along with it, right? So combining together. I'm wondering if that's something that the next steps might be looking at as well.
Yeah, so we're doing the animal studies at the moment with CF10 and everolimus. And I just want to say we also do our studies in male and female rats. I just showed male data just for the interest of time, but we do females as well. But the future plan is obviously once CF10 is up and running, then it'll be SODA and everolimus, because we'll have done the trial hopefully with cysteamine and everolimus, and it'll all be there already.
Was your question about the clinical studies in man? Is that what you were saying with the joint everolimus and CF10 in people?
Yeah. So as Jen says, we will hopefully be teed up with all the preclinical data. There's a catch-22 here, and that is regulatory authorities are really uncomfortable about approving a combination study with 2 unregistered drugs for the indication. Obviously, everolimus is used in the transplant-related setting in cystinosis, but as a therapeutic, it doesn't, to my knowledge, have a license. I'm hopeful that with your support as a community and with the solid data that Jen will generate, we might twist some arms on that one and get them to think out the box. But I've got to manage expectations here. It may be that possibly it would need to be cysteamine and everolimus in the first instance.
Herbie, question for Herbie. The cost of bringing a new drug to market can be expensive and can be prohibitive when it comes to selling that drug to health boards, such as Proxibie that we're finding at the minute. Can you talk around what's being done with CF10 to make it more available?
So, brilliant question, Roy. Thank you so much. And one that we're particularly aware is of considerable interest to the community, given that scars that you carry from Process B. So Ros was always absolutely adamant that CF10 should get to every patient that needs it and that cost should not be a barrier. When you apply to the Medical Research Council for funding, it is mandatory to have a route to market mapped out in your application, because the Medical Research Council get their government money to improve the health and the wealth of the nation, is the mantra. What we've done is set up a spin-out company called Sundara, because that allows us to maintain control over that route. And Sama Taslak, who's the chief exec of that company, was here earlier on, unfortunately had to go this afternoon. Sama, likewise, is not going to see price as a barrier to access for patients with cystinosis. So please, please, please rest assured on that point. Very important, and we're absolutely wedded to this being a cheap and affordable medicine.
Thank you. Are there any other questions?
Yes, is it working? So I had a couple of questions for Jennifer.
Yeah.
So first of all, if now a patient with cystinosis were to get a transplant, do you think there's enough data to recommend everolimus as a first line of treatment for the immune suppression?
I would think so, but I'm not the doctors.
Okay.
So can I just push you a little bit further on that line? Because is there a natural experiment here where we know everolimus or the sister drug sirolimus are used in, not all that often, but in transplantation already? Are the patients with, do we know of patients with cystinosis who are taking cysteamine and have chosen or been chosen for their immunosuppression?
Yes. I'm taking everolimus.
So here we
but
go.
for immune suppression. But it was not the first line of treatment. So I actually started with Mifortic, which is mycophenolate. But I switched to everolimus for different reasons, not because of your research, about 3 years ago. So that was actually my second question, if you have been able to look at data of patients who, because of the transplant, are taking everolimus, and if it might be interesting to look at.
No, I think the data has to be out there. There has to be, like, there's patients with transplants and they have to have what they're taking. And I have tried to get this information, but it's tricky. But I know Stephen, is he here? Looked. Did you look in the American side? You told me.
I mean, I just kind of probed on Facebook to find out who's on everolimus, and it's not a very commonly used drug in renal transplant. So there are like a handful of people taking it.
Yeah, so if we could, like, ideally get like a cohort of people who are already on these and then do some measurements before and after, you know, obviously not before because that's gone, but, you know, from now on and see what the progression is. But yeah.
So I think we could develop that actually. We can go to the European NHS Blood and Transplant and to the scientific registry in the States maybe, just to see if there are, if almost in a way, if that natural experiment has been performed. So I think that's very exciting. We'll be in touch.
And then a third question that's more of curiosity, if you know if you have an estimate if the dose for this case, or not for immune suppression, how it would compare to the one used for immune suppression for people with transplants, if it would be the same dose or lower, because of course we know that this treatment also has other side effects.
Exactly, yeah. And so originally we did choose the high dose of 10 mg per dose, and we've gone right down now to 2 milligrams in our new studies. So ideally, you'd want to go low, and it wouldn't be for immunosuppression. This would be for, you know, combination treatment. So it'd be a different dose to what you get for immunosuppression. So yeah, I don't know how it would work if you're already on it for immunosuppression and then taking a combination.
Yeah. So even at 2 milligrams, that's per kilo,
mostly,
it's still a lot more than...
Mostly in the case that someone who doesn't need the immune suppression because they don't have a transplant, if they take it, not for the immune suppression, but to treat the cystinosis, it would be interesting to know if it would still be the same dose as for the immune suppression.
No, I'd imagine it would be much lower. I would imagine.
No, thank you. That opens an exciting opportunity. And the clinical studies to look how it might work are probably achievable in a reasonable timescale, I would have thought. So, again, moving forward as quickly as we can. Yeah.
Yeah, thanks. Appreciate you might not be able to answer this completely, but I'm wondering what are the timescales from where you are now with CF10 and the clinical trials to a point of it being available for actual clinical use?
So, as I say, we're hoping that we'll be in a position to start the phase 1 trials in 2026. And I know this seems like an archaeological time frame, but please believe me that the Sunderland team and the army of people working on this are just going as fast as we can. The answer to your question will depend upon the regulatory authorities and what they are going to ask us to provide to get the drug registered, and we haven't started that conversation yet. What we do know, we will talk with them on day 1 about, is what are we going to do with patients who volunteer to be on this study altruistically. They start taking the drug for 28 days. It works well without side effects, as we hope very much it will. What are we going to say to those patients on day 29? I'm really sorry, you've got to go back on your rubbish old cysteamine. And the patient's going to say, no, I would like to stay on CF10, please. And so we're going to ask the MHRA that right up front. And those data could well turn into what's called a registration package. And so it's a discussion we're going to have, but just to reassure you that we will be looking for absolutely the most rapid pathway to registration, which will allow CF10 to become available much more widely than it is at the moment, it would be initially.
Thank you, Herbie. David's got a question here.
So actually a comment around the combination of everolimus and cysteamine. So just like Stephen was saying, everolimus is hardly used in transplantation. It's not available in the UK in transplantation, in fact. And also post-transplant, of course, you won't have Fanconi syndrome anymore because you've got a transplant. And that was one of Jen's key readouts was the improvement in Fanconi syndrome. So what will your readout be post-transplant if you're combining everolimus and cysteamine? Well, maybe it's easier to achieve white cell cysteine levels, I'm not sure. So I think it does need a little bit of thought about what patient cohort it should be. And to my mind, and we're talking about this today and yesterday, it would be presumably people with Fanconi syndrome who don't yet have a kidney transplant. That would probably be because that's the unmet need, right? It's the Fanconi syndrome. And if you could give those people everolimus in combination with cystagon or Procys-B, that might be the group to go for. Just my comment.
Yeah, I agree with that. Any other questions? Okay, we've stunned them. And we've actually reached the end of our allotted time anyway. So huge thanks to Herbie and to Jen. Real excitement ahead. The next exciting development, I was just saying, running with Neil, the adult cystinosis group, and one of the issues that we were discussing was eye drops and the ocular complications of, and it's very clear that there is a huge unmet need for further development in that area. And so it's a really great pleasure to introduce Professor Anuj Chauhan, who is professor at the Colorado School of Mines, who's been developing technology, including that of treatment for patients with cystinosis. So Anuj, thank you.
So yeah, thanks for the opportunity to come and talk to you all. Looking forward to your questions because that's going to be very helpful to us as we try to take our project forward towards actually getting those products into your hands. Yeah, the title is a bit complex, but really what we're trying to do is to replace the eye drops with contact lenses. And I'll try to explain to you why we are doing it and why we think that will actually work. So all of you, I think, are familiar with some of the challenges with eye drops. The cysteamine eye drops that you take right now, it could be either Cysta drops or Cystagon. It has poor bioavailability. What it means is that when you put a drop of drug into the tears, the drug goes everywhere in the eye. Now, what you want is, you want the drug to go into the cornea. So this is a structure of an eye where the yellow is the conjunctiva. This blue is a contact lens. I'll talk about that in a minute. And this gray is the cornea, the center part. So you want the drug to go from the tears into the cornea because that's where all the crystals are. What will happen is the drug is not going to just stay here. When you put it in the eye, it's going to go everywhere. All of these tears, they're going to have your drug. And then a lot of the drug will go to this yellow tissue into the blood. And also what happens is every time you blink, you actually push some of the tears into your nose. There are these two holes in the eyes called puncta, which connect your eyes to your nose. So what happens is most of the drug that you're putting into the eye doesn't actually go into the cornea. It goes into the rest of the body. In fact, about 95 to 99% doesn't go. And so that number is really low. And the question is, can we improve that? But because that number is very low, you have to put a lot of drops. You have to put 8 drops or 4 drops, depending upon your formulation is. So if you can increase this number, you will not have to put that many drops. Another challenge is that the drug cysteamine, it can very easily oxidize into cysteamine. And that's why your formulation comes frozen, so that the oxidation doesn't occur. There's also stinging with the eye drops. And one of the reasons for the stinging is the drug itself could cause stinging. But in addition to that, there is a preservative in there called benzochlorium chloride. Because you don't want the drug formulation to develop, have bugs going in it. And to prevent bugs from growing in it, because the air is full of bugs. So when you open an eye drop bottle, some bugs could enter the bottle. And then if there's nothing in there to kill them, they will grow. And you don't want that. So you put preservative in there. And when you're putting an eye drop into your eye, the preservative is going into the eye. And you're doing this 8 times a day. And so there's a lot of preservative that's going into your eye that can cause stinging. It can also actually damage your ocular epithelia. So you don't want that, if possible. And finally, the bottles have to be shipped frozen to minimize the degradation. So there are all these challenges, and depending upon what formulation you use, there are additional challenges, like the Cysta drops are very viscous, they're difficult to put in, they could blur your vision. So there are a lot of reasons why we might want to do away with the drops and go with a better approach. So if you think about what could be a better approach, what we're doing is we want the drug to go into the eye. That's the requirement. Eye drops you have to put again and again and again and again. Wouldn't it be nice if you put something one time and that slowly and slowly keeps releasing the drug? So that was the basic idea of starting this project. And the question was, okay, what do you put into the eye? You could put that product that you want to release slowly at many locations. You could put that in the conjunctiva sac. But a natural device in the eye that hundreds of millions in the world are using are contact lenses. So we thought, instead of creating a new device and that you put somewhere, why not just use contact lenses that are already being used, that have been proven to be safe? Why don't we put the drug into that contact lens and use that as a delivery vehicle? So that was the initial idea that we had when we heard about this problem. And so today I will try to show you, we have come quite far in actually translating that into actual practice. So when that idea came to us, we thought, okay, let's just take a contact lens, soak it in your drug solution, so the drug will go into the lens, and then let's measure at what rate the drug comes out. So when we made that measurement, what we saw that the percentage releases, what percentage of the drug is coming out as a function of time. So what happened is that the contact lenses that we tried, the drug came out very quickly, in about 0.2 hours, which is basically 10 minutes. In 10 minutes, the drug is all out. So that was never going to work, because if in 10 minutes, that would mean that you load the contact lens with drug, you put it in the eye, after 10 minutes, all the drug is gone. So then you will have to put another lens. And the only thing worse than putting 8 drops would be to put 8 contact lenses. So that was never going to work. So we said, okay, so our first challenge is to design a contact lens that will release the drug slowly. So to do that, we came up with a very simple idea. The idea is this. So imagine that this is your contact lens, and these are your drug molecules. So the molecules, basically what happens is when you put the lens, the drug molecules just get out of the lens and go into the tears. Then the idea we came up with is, what if you put barriers in the path of the drug? So imagine there are bricks, and the drug cannot go through the bricks, the drug has to go around the bricks. So with that idea, we tried a few different things, and we are able to successfully create those barriers in a contact lens. So this was the schematic of what we wanted to do, this is what we actually did. This is a contact lens matrix. The picture is a very high magnification picture called SEM. And these bricks that you see, they are actually made up of vitamin E, which is very, very biocompatible. It's actually an antioxidant. It's actually good for your eyes. So that was the idea, to create these bricks of vitamin E, so now the drug will have to go around them, and that will hopefully lead to longer release. Okay, and let's see. So yeah, that's the idea. The drug will have to go around them. And this is just how we make the barrier, so it's not really that important for this talk. So I'm just going to show you the proof that indeed it works. So this is the data. I already showed you the data at the top, where all the drug comes in about 10 minutes. But now, after putting vitamin E into the lens, now here we're looking at AccuView Oasis lenses, which are one of the most comfortable, one of the most popular contact lenses in the world. Like I said, with hundreds of millions of patients already wearing it. And what we have is the percentage release, that is how much drug is released over time. And you can see the one without vitamin E, everything comes out like literally in a burst. But with vitamin E, you can stretch it to whatever time you want, depending upon how much vitamin E you put in. The more vitamin E you put in, the longer the drug is released. And you can see with about 20% vitamin E in the lens, you can release for about 4 hours. And with 30%, you can go to about 6 hours. And that's really what we need. We don't want to release for multiple days because we want a different lens every day. We want disposable lenses. So the idea is you'll load the drug in the lens will go into the eye. When the drug is all gone, you take the lens out, throw it away. Next day, you'll put a new lens. Okay, so this was very promising. So we said, okay, and this is a picture of what a lens looks like. It's very completely clear. It's slightly yellowish because that's the color of the vitamin E. So then we started testing it for safety. As you saw, with any product, first thing you have to do is before you start looking at efficacy, you have to prove the product is safe. So we made these lenses and we put them in rabbits. It was a 7-day study. And during the 7 days, there was no toxicity. I'm not going to go into details of all the studies that were done, but essentially there was no toxicity. And so we said, okay, let's test if the idea works. Now, you might have seen that a lot of animals that were used for a lot of the drug testing are mice. It's challenging to design contact lenses for mice. We actually have designed contact lenses for mice, but it's not an easy study to do. So we had to come up with a different animal model. And some researchers are trying to create a cystinosis model in rabbits, but right now there's no reliable model. So we had to come up with a very simple way of testing whether the drug works. So what we did was that we took, the rabbits are frequently used for eye studies because the size of a rabbit eye is comparable to the size of a human eye. So we took a cadaver rabbit eye. Cadaver rabbit eye means this rabbit was sacrificed for it could be for many reasons, including possibly food, but either way, we get these eyes. And then we put them in a chamber, and then we soak it in cysteine solution. So cysteine essentially goes into the eyes, and then we oxidize it to convert to cystine. So we create cystine crystals. Now, I'm not going to call that as a cystinosis model because this rabbit doesn't have a disease. We're just loading it with cystine crystals. And after that, what we do is, so this is what it looks like. This is simply a picture, high magnification picture to show that indeed the crystals are formed, and the crystals look pretty much similar to the crystals in a human eye. So after that, what we did was we started putting our contact lenses on these animals, or not the animals, but the cadaver eyes, and we showed that the crystals dissolve away. So that seemed promising. So far, based on the data that you have seen so far, it seems that we can design a contact lens to release slowly for whatever duration we want. The lenses are safe in animals, and they also seem to have some efficacy. But the efficacy here is very qualitative. So what we wanted to then see is how much drug is going into the eye that will actually dissolve the crystals. And that's called pharmacokinetic studies. Just like you saw pharmacokinetic studies for CF10, we wanted to do pharmacokinetic studies. That means you put the lens on the eye of an animal, and then you measure the concentration of drug inside the eye and also in tears at various time points. So this is that study. In this case, the concentration I'm showing you is only in aqueous humor. Aqueous humor is that fluid in the eye just behind the cornea. And the concentration is in units of milligram per ml, or microgram per ml. So the first study was with eye drops because we want ourselves to be at least delivering drug equal to 7 to 8 times the eye drop. So we wanted to first measure how much drug goes into the eye of a rabbit with a single eye drop, commercial eye drop. And then we'll multiply that by 8, and that will tell us how much drug we want to go from our contact lens. So this is the data for an eye drop. So the concentration essentially starts with 0 just before the eye drop is put in. After the eye drop is put in, the concentration in the aqueous humor starts to go up. It reaches a maximum, and then after the basic eye drop is completely gone, the concentration starts to go down, and eventually it will go down to 0. So with that, after that, we started putting our contact lenses onto the eye. Now, to put a contact lens, we had choice. We already knew what contact lens we were going to use, but we had to pick how much vitamin E do we want to load into the lens. And that has to do with how long do we want to put the lens on the eye. And that is something that actually we need your input on, and I'll come back towards the end of the talk on that. So we didn't want patients to have to put a contact lens for the entire day, because not all of you need contact lenses. Now, these contact lenses will have power in them. So if you need vision correction, then fine, you can put it for the whole day. But patients that don't need vision correction, to have to ask them to put a contact lens for the entire day just to deliver the drugs seemed a bit too much for us. So we wanted a weird duration that is maybe just a couple of hours, so that you have flexibility. You can do it daytime, nighttime, whatever you want. And so we wanted a weird duration of just a few hours. So that's why we chose vitamin E of 2 hours or 20%. That releases for about 2 to 3 hours, and 30%, which releases about 4 to 6 hours. So those were the 2 loadings that we tested. So first we tested the 20% vitamin E. And you can see with the contact lens, the concentrations are much higher. They still go down, but even after essentially 120 minutes, the concentrations are high. And with a single contact lens, from this you can calculate, and this contact lens essentially delivers the amount of drug that is needed for about 7 eye drops. So we are very close, we're right in the ballpark. And then also with the 30% vitamin E, you can 30% vitamin E, because there's more vitamin E, the drug comes out slower. So the concentrations are lower, but will last much longer. And so in that case also, we can deliver about the same amount of drug. But the reds, if you put 30% vitamin E, you'll have to wear the lens for longer duration, maybe about 6 hours. But if you put 20% vitamin E, you may have to wear the lens only for about 2 to 4 hours. All right, so in addition to that, in addition to measuring the concentration in the aqueous humor, we also measured in different tissues. Cornea is where all the crystals are, so you wanted to measure concentration. And you can see even in cornea, with these vitamin E loaded lenses, we can actually get higher concentration compared to eye drops. Retina is the tissue in the back of the eye. There are still some crystals in the back of the eye, so it's still good to be able to deliver some there. And they'll also be able to deliver at pretty high concentration. So that was good. That means I think we are on track to essentially, well, this is just a mathematical model, so I'm going to skip that. So far, everything looks good. And that means the next step, typically after animal studies are successful, is to go into humans. But before you put the drug into the humans, we just wanted to test the vitamin E. That are the lenses with vitamin E, are they safe and comfortable? So we did a human study with 10 patients where we put the lenses on the humans for 2 days. One day they wore it, and then they changed it and wear another lens. And there was absolutely no toxicity. Patients are completely comfortable. We add a lubricant to this contact lens to make it very, very comfortable. So these lenses are actually very comfortable, very lubricious. So we thought, okay, now we can start putting drug into the lenses. Now, we haven't done any clinical study so far with cysteamine lenses, but we've already done clinical study with other drugs. So these lenses have already been on patients, including a drug. So the drugs that we have tested are glaucoma drugs as well as anti-infective drugs. But for cysteamine, we haven't tested it, but nonetheless, we are ready for it. So to start doing human studies, you have to first get what is called an IND approval in the United States. It's called Investigative New Drug Approval. We already have received the IND approval from the FDA. That means the FDA has told us, we believe that your product is safe and it is fully validated in terms of manufacturing. You can go ahead and do the study. But in order to do the study, of course, you also need funding. So we have already submitted a proposal to the FDA. The proposal was reviewed reasonably well, but we have to submit some revisions. So we are hoping that we're going to submit those revisions in October of this year. And so October of 2025 is when we hope to get the funding. So we hope this study will start in 2026. Again, I wish we could do this faster, but yeah, the timelines for clinical studies is fairly long. But I do want to describe the clinical study to you because I think we are at a point where we can make some changes to the study. And that's what we want some input from you to see. And I'll tell you the exact questions on which we want your input to see what do you think, how should we do the study. So the idea is that we'll start with with the consent. So the patients will come and we'll test their eyes to see what their crystal density is. So when you go to an ophthalmologist, typically the ophthalmologist uses what is called a slit lamp to look into your eye. From the slit lamp picture, they can calculate how much crystals there is. And that's called a crystal density. So the crystal density score will be obtained. And then for 2 months, we'll do what is called run-in. That means you'll keep using what you're already using, just so we get a good baseline. After 2 months, we're going to put the contact lens in you. We're going to have 2 different groups, a low dose and a high dose. And this study will go on for 10 more months. And during this study, you will be essentially putting a new lens every day. One question we want your input in: how long do you think you should wear the lens? And there's a survey for that. And we'll give you this link for the survey later. So that will be one question that is asked. Another question is that while this study is going on, would you like to put lenses in both eyes, or a lens in one eye and eye drops in the other? The benefit of putting lenses in one and the eye drops in the other is that you have what is called a control eye. But the downside for that is that it might be that the patients have to essentially still use 2 different products, and so it'll be good to get your input on what do you think you prefer. But either way, then we'll have the study for 10 months. After 1 year, the first phase of the study will be over, and then we will essentially assess and see which dose was more successful. And depending upon which dose was more successful, we're going to do what is called an extensional phase. In the extensional phase, you just use a more efficacious dose, and hopefully the entire study will conclude in about 3 years. And so 3 years after starting, we might go to the FDA and try to get approval. So that's essentially, this is the inclusion and exclusion criteria. The only important question here that I think we are still debating about is the age of the patients to start with. We don't want to give contact lenses to very young kids, although ophthalmologists say that a kid as young as 4 years old can use a contact lens. And ultimately, with the approval, we think we will go lower in age. But for the initial clinical study, we want to stay at about 8 years old. So we started with 12, but now we have decided to actually go down to 8. So this study will be available in the United States to kids who are 8 years and older. So just to summarize, we have developed a product that can sustain the release of drug for long periods of time. We have shown that product is safe. It can dissolve crystals. We have already proven everything in animals, and now the human studies should hopefully start once we secure funding, because the FDA has already approved the study. So with that, I would like to thank you all for your attention. Willing to help questions.
Anuj, thanks very much for an incredibly clear and inspirational talk. Do we have any comments from the audience? Yeah, down here.
Thanks, Anuj. Thanks for the great work and also the great presentation. My question is, what is the strategy for not using the preservatives with this pathway of using a lens so it won't sting? Not using the preservative, which causes the eye irritation.
Great question. So this contact lens will not have a preservative. The reason you need preservative in your eye drops is, with the eye drops are still in your bottle, it's sterile. It doesn't need a preservative inside because it was made sterile. The preservative is just to be there once you open the bottle. Now contact lenses, once you open it, it will go into the eye, and the next day a next lens will go into the eye. So because it's a single-use, single-day lens, therefore there's no need for preservative. Inside it's already sterile, so there's no preservative, because inside you don't need preservative because it was made sterile. And once you open it, like I said, it goes straight into the eye, so there's no need.
Yeah.
And Joel, thank you so much. Is the downstream idea here, or let me just ask it like this: the patients that already have a prescription for eyeglasses, right? It's contraindicated, for them to have these eye drops and then throw contact lenses over. So I found in our population that most, if they do have, less than 20/20 vision, and it's going to be glasses, would this set of contact lenses, could they actually be used for those that do not have 20/20 as well?
Yeah, great question. So these contact lenses ultimately will, and Rachel, please feel free to jump in. Dr. Rachel Bishop is helping us with the clinical study, so feel free to jump in if you want to add anything. But yeah, these contact lenses ultimately will be designed to provide vision correction as well. Now, the reason contact lenses are contraindicated with eye drops right now is because you will have to remove the contact lens, put an eye drop, wait for about 5 minutes and put it back in, because you can't put the contact lens, you can put an eye drop on top of the contact lens because the preservative in there actually can go into the contact lens and cause toxicity. So yes, ultimate approval, these lenses will be approved for vision correction. They could also have photophobia part in there. So they could actually block some light and they could also address photophobia. However, for the clinical study, we are not going to have power in the lenses for multiple reasons. Rachel, do you want to maybe address that?
Yeah, so thank you for making it so clear. One consideration is most patients with cystinosis don't wear contact lenses. As you're aware, we don't really know how well contacts will be tolerated. Sometimes with the cornea already being affected with crystals, it's possible that wearing a contact lens for 2 hours is quite comfortable, but wearing it for 4 or 6 or 12 or 16 is not. I personally don't tolerate contact lenses just because I have dry eyes just because. So for some people, a contact that is intended for a whole day wear may never be something they really want. That being said, this contact lens is prepared to be your vision correction if you end up being someone for whom contacts are comfortable. And a lot of my patients, this is just patients overall, not patients with cystinosis, I end up saying, look, wear contacts as long as they're comfortable. When they get irritating, when you feel dry, first of all, you can use lubricant drops, and that goes for even when you're not wearing contacts. If your eyes feel dry, non-preservative lubricant drops are a very good treatment. Anyway, some people will find, hey, I like to wear the contacts for 6 hours, that's my comfort zone, and then I switch to my prescription glasses. You can be a hybrid user. There is no problem with that. So the question of will you want to wear contacts all day, I can't answer that. That's not a study that we've done in patients with cystinosis, but happily, this is an option I believe we'll have going forward. There are quite a few reasons why it makes sense to not have that be the test plan for our first group, because logistically, we want to separate the treatment effects, the results from if somebody is wearing a contact all day long and therefore having a lot of discomfort, is that because they're just not suitable to contacts for 16 hours or because something with the drug irritated their eye? We don't want this confusion. So we're going to answer these questions kind of separately. Does that help? And I will just add, I'm quite aware that we are here in the UK, and this study, of course, is being developed, hopefully, to run forward in the United States, and sensitive to the fact that you can't easily hop on a plane and be part of this study as much as we'd love to be able to say you could. However, as you know from drug history, were this to succeed, which we hope, and it comes here eventually, of course, this will be a study that we will be asking you to then be part of. And in terms of survey responses, giving your thoughts to some of these questions we've put on a survey, which we'll offer later, please, this is just, if you're motivated to give some input, we appreciate it. If you're not, there is no pressure. We just appreciate getting, being able to share the research and then getting some feedback from those who would like to. And I'll share more about cystinosis and the eyes tomorrow when we talk about how best to manage with current therapies.
Thank you very much. I think we've got another question down to the left here.
Hi, some of my questions were answered there. One of them was to do with when or how can we get involved, but I'm guessing as a UK citizen, for now it will be in America based mainly. The second question was also, could this, I was aware, as my understanding, I've been told before, wearing contacts could be quite difficult because of the crystals in the eyes. That was my understanding. Is that, like, how would you get around that? And could it be, if you're saying could be worn for a few hours, could potentially be used in conjunction with a drop? Perhaps you wear for a few hours in the day and then a drop in the evening if you can tolerate for long periods.
So excellent question. My hope is with the amount of drug being delivered by this contact lens, as I anticipate, I do not think you would have to use cysteamine drops in those other hours of the day. You might need lubricant drops if your eyes feel a little dry, but the hope is this is a complete replacement for cysteamine eye drops.
Thank you.
I'm presuming then the lubricant wouldn't affect the cysteamine.
That's correct. So in general, what we say with eye drops of any sort is when you put a medication eye drop in the eye, you wait 5 minutes before you put a lubricant drop. Let the eye have a chance to absorb. And that philosophy gives you a very good demonstration of how much time we actually think that drug is having contact with the cornea. I will add, and this is just in the hypothesis zone, it's quite likely that wearing this contact lens even for an hour of continuous drug exposure to cornea is way more interaction with the cysteamine crystals then putting drops in even 16 times a day. If it's 5-minute contact time, even 10 times a day, 50 minutes, we don't know these answers, but it's quite possible that this constant dosing actually means maybe you wear it an hour a day eventually when we understand it better. You may recall the cysteamine eye drops that came out as Cysteran in the United States in 2012 were studied hourly while awake. Right, so the FDA approval label says use hourly while awake, 14 times, 16 times a day. Of course, no human can really do that. So we have to pick something and we start from there. And perhaps we find out, hey, you wear this for 6 hours every 3 days, or I just don't know. But our starting point is the best guesses we can make at this time.
Thank you very much. I'm going to have to call this session.
Actually, if I can just show the last slide about acknowledgement. Sorry, I forgot that. I really want to acknowledge Cystinosis Ireland for providing a lot of the funding for this work. In the last cycle, they also joined the Cystinosis Foundation UK. So yeah, thank you for the support.
Thank you very much, Anuj. Incredibly exciting. Now, the next talk, if I'm correct, looking at the IT people here, is actually a video. Is that right, from Dr. Paul Gaudieu? Am I saying a yes? And so that should appear on screen. Paul was due to join us here. Many of you will know Professor Gaudieu. He's Professor of Pediatrics at McGill in Montreal. But you'll also have heard of the cyberstrike issue last week, which influenced all of the routing of the aeroplanes, and he was caught by that. So it does demonstrate how much of an integrated world that we live in. And Paul's very kindly agreed to produce his talk and is going to show it.
Now, the motivation to embark on this is, I think, captured nicely by this slide from Dr. Brodin Sartorius 12 years ago. This was a study in which a group of people with cystinosis were their natural history was studied. And on this slide, what you can see is that on the y-axis, the left vertical axis, is plotted the age at which an individual with cystinosis develops end-stage renal disease, ESRD, or time for dialysis or transplantation. On the x-axis beneath is the age at which an individual started cysteamine therapy, oral cysteamine. So if we focus first on the right-hand side of the slide, in the lower quadrant, this is a group of people who never received any cysteamine. And if you let your eye drift to the left over to the y-axis, you can see that they went into renal failure at about the age of 10. Not very good. Now, if you look at the left upper quadrant, marked in blue here, you can see that there are a sizable number of people who started cysteamine therapy before the age of 3 in this study. Those people really had a big benefit. They went into renal failure not when they were 10, but in their 20s. And these people at the top of the bar here are open circles. They have not yet gone into renal failure. They're up to 28, 29 years of age. So fantastic. This is a therapy which has made a big difference for everyone with cystinosis, and it was discovered 40 years ago. Now, the disappointing part to this therapy comes from the fact that even though these people clearly have had a benefit, nevertheless, they are projected to go into renal failure at some point. So it's not a cure, it's a delay, a partial improvement. And the other observation we can make from these data are the fact that in this red circle on the left lower part of the graph, you can see that about a third of the group who, although they started cysteamine therapy before the age of 3, they have not benefited very much. And the average age at which they're going into renal failure is very similar to those people who never saw any cysteamine. So we need more. There's something we're missing. One possibility is simply that the people who don't benefit from cysteamine therapy are unable to tolerate it, and so they don't get enough of the medicine. That's possible. But scientists have suggested, a lot of research has indicated that there may be some other explanations. So I'd like to go after those explanations briefly with this little cartoon. So this is a picture of a real proximal tubule cell, I think from a mouse. And you can see those little finger-like projections from the cell projecting upward into what is the urine flowing by. And one of their jobs is to capture proteins that have drifted into the urine and not let them escape. They're all good nutrients. And so those little finger-like projections will capture them, encase them in a small vesicle, and send them down into the lysosome. They're chopped up into the amino acids. The amino acids get out of the lysosome and get back into the cell to be used to build new proteins. And there's another system depicted on this cartoon labeled AP, and that is also a set of small vesicles that actually go around the cell, cleaning up debris, unused mitochondria, like these dark structures in the cell, damaged mitochondria, or proteins that are no longer functional. They gobble them up and they, again, come to the lysosome, dump their cargo, and then leave. And again, those proteins are chopped up. So there's a huge amount of cysteine, among other amino acids, that is produced inside the lysosome. And the cysteine, like all the other amino acids, has to get out to be used. And the trouble in cystinosis is simply that the gene encoding that little channel is faulty. Now, cysteine builds up, destroys the lysosome, destroys the cell, so it's not good. And what happens with cysteamine is that cysteamine gets inside the cell, inside the lysosome, chops the cysteine through a chemical reaction into 2 parts, and each of those smaller little subunits can get out through another alternative channel, the back door, so to speak, even though the primary channel is messed up. So that's the way the cell works. The other fascinating thing, unfortunately, is that there seem to be functions for the protein encoded by CTNS, which are not just the channel that allows cysteine out of the lysosome. These other functions seem to be necessary for the capture of proteins that I've told you about and the capture of debris from inside the cell. And those processes, if cysteine is not there, are faulty, and there's nothing we can do about it by giving cysteamine. So that sort of thought process has always made me think that in the end, we're going to need something to put back the normal cystinosis protein that is needed for many jobs. And when I need a bit of inspiration, I simply look at this picture of a bunch of kids, amazingly delightful kids, at a cystinosis meeting down in the United States, and I envision that they're all smiling, but they're probably saying, "We're hopeful. We're hoping for a new leap forward. It's been 40 years, after all, since cysteamine was discovered. We need something more, and we need to find it." So, my low point, perhaps, in this long time journey of my own was during the COVID pandemic. And my research program had to stop, and one of our projects was completely halted. And of course, for all of us, research or no research, for all of us, the pandemic brought sort of a bleak future. Sort of like this picture of the Western Irish coast on a bad day. Nothing really brilliant to look at. But across from where you're sitting in Manchester at the Manchester Art Gallery is a picture of this fellow, John Milton. He was a famous English writer, and he is said to have penned, among many other things, the phrase, "Every cloud has a silver lining." And of course, he was referring metaphorically to the sunlight wrapping around the edges of this dark cloud, indicating that there was something better coming along. Now, for me, the silver lining to the pandemic came in the form of this fellow, Paolo Martini, who was the chief scientific officer for rare diseases at Moderna. He came to McGill to speak to us. And of course, he talked about the mRNA technology that was used for the COVID vaccine. But he also mentioned his idea that it could be adapted to treat rare diseases. I had the fortune to have dinner with him. And of course, what I said was, well, so could we use this for cystinosis? And he said, why not? There's some work to be done, but it's possible. Now, what was the crux of the idea? As you know, I'm going to do a little diversion for a moment to explain how the COVID vaccine was produced. And as you know, our DNA contains genes. Each gene has the code that can be copied onto an RNA, which then produces the protein. Now, the Moderna scientists and the Pfizer scientists knew the code for these little spikes that stick up from the COVID virus, the spike proteins. And so they plugged them into their computers, typed out the mRNA code, and then they synthesized the RNA artificially, stuck it into a lipid droplet called a lipid nanoparticle, LNP. And they took that mix of things, shook it up in a vial, stuck a needle into the vial, and pulled a little bit out and shot it into our arms. And that was the COVID vaccine. Now, what happened in the minutes and hours and day or so to follow was that stuff floated around in our bodies. And when it came in contact with certain cells, the cells would gobble up the LNP, bring it inside the cell, cause it to open up and discharge its RNA. And that RNA then was available to bind to the work table for these things, the ribosome, and produce the protein that was encoded on that mRNA. And of course, in this case, it was that spike protein. Now, when the immune system saw that, the immune system would say, "Oh, this is foreign protein. This is not right," and form an antibody against it. And that was the basis for the protection. How would this work for cystinosis? Well, in somebody with cystinosis, nephropathic cystinosis, there would be two the copies of the CTNS gene, and both would be faulty. So they were unable to make the proper RNA to make the protein that would keep the cell healthy. So I proposed to Moderna that they would punch out the proper code for CTNS mRNA, stabilize it in their special way that does not change the protein at all, but makes the RNA last longer, package it inside an LNP, shake it up in a vial, stick it into people's arms, and hope for expression of CTNS. The idea is that the particles would be taken up by our cells, released, the RNA released, and when this RNA gets onto the ribosome, it doesn't make COVID, of course, it makes CTNS, the cystinosin protein that we need in our cells. Now, can this strategy really be applied to humans? A major leap forward really came from the Moderna rare disease group themselves. This is a paper reported in Nature about 4 or 5 months ago, and it was an interim analysis of their first human clinical trial. They were studying the mRNA therapy that was being given to people with a rare disease called propionic acidemia, not cystinosis. But this clinical trial began back in 2021. They actually used 2 different RNAs to produce 2 subunits of this enzyme that was needed. They recruited 16 people, kids. They followed them for up to 1 year, and they gave them different amounts of the RNA. And they had 15 centers spread across Canada, the United Kingdom, and the United States. So this appears to be promising. We haven't seen the final result, but it's going along and seems to be going smoothly. So there's hope, I believe, on this basis. So I decided to go back to Moderna, rediscuss things with them, and Paolo Martini put me in touch with this lady, Megan Brundage, who's a scientist at Moderna. And so she helped me get this ball rolling. With Moderna's support, I then applied to the Cystinosis Research Foundation in California, and was awarded a grant to pursue this. And the funding began in January. So this is relatively recent. The first job I had to do was to figure out the sequence that I would ask Moderna to produce. And so shown here in the blue circle, the classic cystinosin protein, or the RNA code, is very well known. And this is the one that winds up in the lysosome, in the membrane of the lysosome, and acts as a channel to get cysteine out. No question we had to supply that sequence. But to make things a little bit more complicated, thank goodness, long ago, Anna Taranta and Francesco Emma from Rome discovered that there was at least one other form of cystinosin protein, or CTNS mRNA, and it had a longer tail on it that stuck into the middle of the lysosome than the usual one. Everything else was the same, but it had this long tail on it. And it looked as though this accounted for 15% of the total cystinosin protein in the cell. And it seemed to me that it was foolish to think this was doing nothing. And so I decided at that time that we really would have to consider both mRNAs as part of therapy until we knew more. And in fact, Anna and Francesco had spotted the fact that this other form of cystinosin appears a little bit in the lysosome, but also very prominently at the surface of the cell, and associated with some of the little vesicles spinning around in the cytoplasm, some of which may or may not be the particles that are transporting proteins from the surface or transporting debris into the lysosome, as I mentioned before. So I just felt that we could not ignore this and take a guess that the main form was the only thing that would be needed. So I went back to Megan Brundage, explained to her the whole thing, and said, we need you to make an mRNA for both forms because we need to test them both. Furthermore, we need to follow them when they get into cells, and we need to be able to watch over them. So can you stick on some tags? And she said, no problem. I've got plenty of little tags I can type into the sequence and put them at the beginning or the end or wherever you want. And you will be able to track the protein product that comes from the RNA that we're going to give you. I said, we'll take them all. We'll test them all. So she sent us the first RNAs in March, and we began to test them in April. So what we did is outlined here. We took those RNAs that Megan had sent us with little tags on the ends, the two different types of cystinosin and a little tag on one end or the other, and we artificially just forced them into the cells in the laboratory, and we used human kidney cells from the proximal tubule. These cells are very well known to everyone, and we use them to do the job. So we then began to encode. Well, we then we smashed up the cells. We took the proteins that were inside those cells and spread them out on a gelatin slab. And then we exposed that slab to an antibody against the tag, V5, so we could find our protein wherever it was. We could even figure out how big it was. And sure enough, we found what we wanted. We found a main band right at the right size, and it reacted with an antibody to this tag, so we knew that was the real stuff, the real McCoy. Then we did the same experiment and said, well, we know it's the right size and it's the right protein, but is it going to the right place? So this time we analyzed the cells with a microscope. In this slide, we're looking down through the microscope to see the red color that represents our little tag on the molecule. So when the molecule is produced, we'll be able to see this red tag. And there they are. They're little blobs of red inside a single cell. But is it really in lysosomes? And the fact is that they are, because we stained the same cells with another colored antibody that would detect something on the surface only of the lysosome. And so if you look carefully, every blob of red corresponds to a blob of green. And you only see the red stuff where you see the green stuff. So this does exactly what we wanted it to do. It goes right to the right spot and nowhere else. And just as predicted by Anna and Francesco, the other isoform, the other form of cystinosin with this long LKG tail, appears at the surface, that line running along the outside of a cell, as well as in some of the little vesicles inside. So this is exactly what was spotted before. And so we think that both forms of the cystinosin that was produced by mRNA, the CTNS mRNA, both forms are correct. Sorry about that. So one final problem. We think we have the tools, the mRNA tools, to do the job, but there's one more task that is an important hurdle, and that is that the LNPs, the delivery system for these mRNAs produced by Moderna, they love to go to the liver. The cells of the liver just gobble them up. Nearly 100% of the LNPs get into the liver. And the problem is that the liver is not much of a problem in cystinosis. We really need to see the RNAs delivered to the kidney, the muscle, and the endocrine organs. So that was a problem. And I began talking to Moderna about this a year ago. And they, just 1 or 2 months ago, sent me this information. What they did was to take their standard LNP, lipid nanoparticle, and they made RNA for this fluorescent protein, GFP. And so they packaged the RNA in there, and then they injected it into a mouse. They took out the kidneys, and they did this analysis. And what we're looking for, forget this big red blob and that green blob, we're looking for another green blob that appears at a smaller size, which is expression of GFP. And oops, the standard LNP that worked beautifully for making a COVID vaccine doesn't deliver to the kidney. They had other formulations that they tested. So LNP2, nope, there's nothing. They tried all sorts of tricks and nothing in the kidney. They tried another one. They had some new ideas, but no, nothing at all. And then they came across a most recent novel, a new formulation of the lipids and things in the coating of this lipid nanoparticle, the amount of cholesterol and so forth. And it was a gold mine. They found that here's that signal that we're looking for, that's GFP. And it was seen only when the mice were injected with this formulation, which I call now, we agreed to call Charlie, because that was the name of the dog for one of the Moderna scientists, Mike Zimmer. And so we agreed just to refer to it as Charlie, because Moderna does not want to tell me exactly what's in there and what the name of it is, their name. So, but in speaking to Mike Zimmer, we agreed that this looked really good. We're going to package in the future the CTNS mRNA into this thing, try to get it to kidney, and then we're in business. So with that, I'll quit. I think that that this project looks very interesting, and it looks like this fjord down in the bottom of the South Island of New Zealand. It's so interesting. It appears to hold such promise that we just have to go in, take a look at this, and see what's there. It was actually a fjord that Darwin sailed into. He discovered some flightless birds and some other stuff around the corner of this very fjord, and it helped him to develop his whole theory of evolution. And so I hope that our foray into this fjord, or metaphorically, will bear fruit. These are the people who are actually doing the work in my lab on this project. These are my collaborators. My wife, Elena Torban, is actually a co-PI, principal investigator on this project. Mike Zimmer and Megan Brundage are from Moderna. I talk to them about every 2 weeks. And of course, this would not be possible had it not been funded by the Cystinosis Research Foundation. So I thank them for taking a chance on this project and funding it. I hope that it will deliver in spades. Okay, thank you very much.
So another major element of hope for the future, and it's fascinating how advances in one area of medicine and science actually can have major impacts potentially going forward. So that takes us to our final speaker for this session. And we're moving from the laboratory and elements of science to something that's equally important but more immediately deliverable, which is delivering the service, the care for patients, the organization of service delivery that really we want to deliver, the one that's optimal for patients with cystinosis. And so I'm going to introduce Dr. David Game. David is a friend and colleague who, here he who is a consultant nephrologist at Guy's and St Thomas's Hospital and has been instrumental in the development of an institution of the national designation of specific cystinosis centres within the UK. So, David, thank you.
Thanks very much, Graham. So, some fantastic talks just then. I don't know if everybody else's scientific brain is a bit saturated with science, but mine definitely is. And I think if you followed all of that and you're still going, then you could probably answer at least a degree level question on molecular biology. So it's really great talks and very illustrative and some really inspirational work for the future. So don't worry, I'm not going to talk about mRNA or anything like that, even if it's your thing. I'm going to talk a little bit about how the organisation of care has changed in England. And I'm approaching it from a fairly adult side of the equation because I'm a nephrologist for grown-ups. But obviously this involves paediatrics as well. So I'm going to talk about these national designation centres, but I'll talk about the word hub or hub centre or multidisciplinary clinic. I'm meaning those things are all interchangeably. They're going to be the same thing. We're going to talk about how these hubs were set up in the first place, how they look now, a little bit about transition between paediatric and adult services. I'm going to tell you a bit about radar if you don't know about that already, but I'll tell you about that. And then some thoughts about how we might take these hubs forward. And I'm not going to take the whole half hour, so you'll definitely get to your coffee in good time. So a big shout out to Cystinosis Foundation UK, who were involved from the very beginning of this, around 10 years ago, believe it or not. And that's Jonathan Terry. I don't know how many of you here knew Jonathan. He passed away a few years ago, and him and his father were instrumental in setting up the foundation. As you can see, he had cystinosis and had an MBE for his services to charity. So nobody in this room needs persuasion that people with cystinosis would probably benefit from going to a one-stop clinic rather than going to a million different clinics, one after the other, well, actually not after the other, different days of the week, different hospitals, different specialists, some of whom don't know that much about cystinosis. And so we decided we'd try and put some thoughts together. And those of you that have commented on how long everything takes will see what's happened here. And it was 10 years ago that we had a meeting of a few people in England. I think it was in Birmingham, and we kind of had this core writing group and wrote a few things together. And you can see all the different hoops. And Elena talked about all the problems in trying to get drugs through to market these days, and it's not dissimilar to clinics or anything that you want the NHS to pay for. It's really very difficult, and they put lots of barriers in the way, probably intentionally, I don't know, and you have to just go through each one methodically and try and persuade people that you're doing the right thing. And so you can see from here, from the core writer group in June 2014, then we had the consultation, the service specification, which I'll tell you about, and then then you get to the tendering process, and then you put your tender in. And so actually it wasn't quite 10 years because it was autumn 22 that the first clinics were officially started. And we've hopefully made some progress since then. So sorry guys, it just takes a long time to do stuff in healthcare. And this is the model that we based the whole thing on. This was drawn up fairly early together with NHSE. And what we're trying to show is that the hub, it's a hub and spoke model, right? So the hub is where the adult and pediatric sort of centers are. It's a multidisciplinary clinic, so hopefully just one stop for everybody. We've got everybody, hopefully, that you need to see in one stop, but actually the delivery of quite a lot of this, maybe all of it, depending on where you live, will be local. And the idea being that you identify the need here, you cajole the local people to do what they need to do, and then try and follow up to make sure that that is actually happening. Obviously, you'll have other specialists involved quite apart from the MDT. Clinical genetics will be involved in parts of the journey. Obviously, we need to measure the white cell cysteine, and then there's preconception counselling for some people, and other services that will be brought into the whole model. And everybody should be having at least an annual review in the hub. That's what we said we would do. Some people have more needs than others and are seen much more often, and others have very few and will be seen every year. And so the specialists that need to or should be at the different clinics will be different between adults and children, obviously. And so this was the list that is drawn up in the service specification. You can see the whole thing online if you want to. It's a kind of a contract between the NHS and the hospital that's delivering the care. And so you can see that so in PEDS, for example, you'll have some genetics, for example, that may assist with the diagnostics unless you're also using white cell cysteine. Pregnancy in pediatrics, probably less required. Pharmacy for everybody. Physiotherapy, it's definitely important in adults because the muscular syndrome, if you like, is more common as you get older. And so different things required at different times. And so that's what we are supposed to be delivering. And so the tenders went out, the hospitals put their bids in, and the way it was formulated eventually by the NHS England was that you had to put a bid in jointly between a paediatric centre and an adult centre. So when they first put it out, it was going to be a bit of a free-for-all, but then they said, no, we want you to go in together so we can ensure that the transition process is good. And so naturally, we went in with the Evelina in London because that's based at my hospital, and then Birmingham and Birmingham Children's, and then Manchester Children's were also awarded the contract. And so I've given some pictures, the nicest pictures I could find of the hospitals, Guy's and St Thomas's are two hospitals, but this is St Thomas's Hospital here, and that's where the Rare Disease Centre is, where we have our both adult and paediatric clinics. And the idea is that every patient in England is under follow-up at one of these clinics, realizing that, again, most of the care in a lot of cases will not be primarily delivered by that hub, but that hub will have oversight of all of the medical care that we think you deserve. And that's the sort of the mantra, that's the plan. Patients from outside of England can be seen in these hubs, and there are commissioning arrangements for that. So that can happen, although Scotland has a very good nephrologist in Glasgow who runs a cystinosis clinic there. And so, as mentioned, really, the purpose of the hubs is to support patients with cystinosis, their families, carers, and local clinical services to ensure equity, really, so everybody's getting high quality care, because that was what we observed. Some people were getting a great service and other people were getting a less great service, and we want everybody to get a really great service, at least as best as we can. And so, as I said, everybody will have annual review, and if there's trouble with transport, there are mechanisms within the NHS to be reimbursed for transport to clinics. So just some pictures of the Rare Disease Centre at St Thomas's Hospital, and I know Graham has a Rare Disease Centre where he holds his clinics as well. And the idea about this sort of centre is it's a little bit nicer than your average transplant clinic or your average kidney clinic, right? So it's supposed to be a little bit more aesthetically pleasing. Sometimes we can get a little bit of tea and coffee if you're lucky, and sort of more information about your particular condition, in this case obviously cystinosis. And then the Rare Disease Centres are usually set up with lots of different clinic rooms. We've got 8 different clinic rooms, and so depending on what rare disease is, then either the clinician sits in the room and the patients go in and out of the different rooms, or the patient sits in the room and the clinicians go in and out of the rooms, depending on what which model really works. And that's what we do. And we kind of try and work out which specialists each patient needs before they turn up, because not everybody needs to see everybody. And so just some sort of pictures of MDT working. So this is the picture of the eye clinic. So our eye clinic is just downstairs from the Rare Disease Centre. So a guy sort of walks people down, they get all the eye assessments, and they come back up again and see the other specialists. That really works really well for us. Physiotherapy can come up with some really novel devices sometimes for people with muscle weakness, and that's really great. This is what's called a SNP test. This is measuring respiratory muscle strength, and that really became pretty. I don't know who's done a SNP test in this room, but you got to put this thing up your nose, and then you got to breathe in really hard whilst blocking the other nostril to measure how strong your respiratory muscles are, and it's quite hard work. And George, my colleague, is a Greek guy, and he goes right, and he goes right, ready, and go, 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 and then the person goes. And trying to breathe in really fast to try and show the bigger strength of their respiratory muscles, and so that can be quite something. But I think it's important to be checking these things, because we would like to think that if we detect that there might be a problem coming along, that we can intervene and hopefully make it better, or at least slow any sort of process down. That's an MRI scan, and I have a fairly low threshold for doing an MRI scan of people with cystinosis for several reasons, but mostly because we want to reassure ourselves that the pressure in the brain is okay. So the hubs provide specialist medication, so you've heard a lot about that today, and so the hubs will be, we can arrange to have Cystogon and Cystodrops delivered to your house. We can arrange for prescription of other medication, including the Fanconi stuff, and also for the patients that don't do well on Cystodrops, we can try and help you with the Cystodrops, or prescribe the preservative-free stuff from the aseptic pharmacy. RaDaR is a rare disease register, which I've got a slide on in a moment, and we submit data to RaDaR, more data than is required for the basic registration of people with a rare disease, because we're using this as kind of our database for follow-up. And I said that hubs are integral to further applications to NHS England, such as proposals for other treatments such as ProCysB. So in terms of transition, so moving from peds to adults, children will, under follow-up in a paediatric centre, may well transition to their partner hub unit, for example. So our paediatric clinic is in the morning and the adult clinic is in the afternoon, so you just got to stay for lunch and then come to the afternoon clinic. But if you're going off to university somewhere else, for example, or college or something, then you don't have to stay at their own centre. You can go anywhere, really. So if you happen to be leaving London and going to Manchester, then you can see Muir in his unit instead. We really don't mind. Adults who are not currently under a paediatric centre can then basically go wherever they want. We don't mind. We won't take it personally. If you live in London but still want to go to Birmingham, that's up to you. We really don't mind, so long as somebody is seeing you. And recruitment is a bit of an issue, so if you're not currently under a hub centre and you want to be, then we can easily sort that out. Letters were written to all the chief executives of hospitals, and any of you who works in hospital will know what happens to that letter. It sits in their desk, and then it gets emailed to various people and cascaded, and nobody reads it. And so that's not the best way to try and recruit patients, I would say. And referrals are by conventional email at the moment, but I've got another slide with some more emails on it. So transition for any kidney patient, actually, but particularly with cystinosis, can be quite difficult, because even though kidney care is delivered by paediatric hospitals, adult kidney care is delivered all over the place. And so if you're transitioning from, let's say, the hospital in London, paediatric hospital in London, you're having your dialysis in central London, but you live in Brighton, then your dialysis will be delivered in Brighton. And so there is all manner of transition arrangements that has to happen to make your healthcare work better. And so we want to make that better by more formalizing the transition processes in all the different specialties that require a transition in. So if you've got lots of different health needs, then those will have to be transitioned to your local services, but under the sort of watchful eye of the hub to make sure that everything is happening. And I accept that historically we sometimes have struggled a bit with that, but we're aware of the problem and we know what we think we need to do. So then what did we do? So do we know that we're doing the right thing sort of compared to the international audience? And I realized that this is an international audience. So we compared what we did are doing in the hubs to what the best guidance available is. And this is a review published a few years ago that talks about all the different specialties that some people with cystinosis need to see. And so for this audience, I'm not going to go through all the different tables, but I basically mapped out all the things that we do and are we doing everything that they said we should be doing? And the answer is almost. So we don't have dental care in our clinic because that's impossible in the NHS. And we actually don't have dermatology in our clinic, but most of our patients, in fact, have kidney transplants and would be seen anyway, and we can refer if needed. This is a website that anybody in the UK probably should have a look at because it's got some good information about the hubs and other things regarding cystinosis. It's hosted by the UK Kidney Association, and it's got patient information, clinician information, and then a dedicated page for information about the hubs. What you will see on that page is some contact details for the different hubs in England. And we've all agreed that if you haven't been referred to a hub, ideally it should be from your clinician, but if you're struggling with that, then just email us directly. Just put rare renal into Google, this will come up, and just email any of us, and we'd be more than happy to help about our own hub or anybody else's. We're really here for you. So at the moment, and these are the numbers of patients under follow-up you can see in the different units. There are cohorts of patients who are still under other hospitals that we all know about, and we're just pulling those patients into the hubs at the moment. So these numbers, next time we talk about it, hopefully will be quite a lot more, and then hopefully everybody will be under review. And through radar and the number of prescriptions for Cystagon that are offered, we know that there are quite a lot of people who aren't currently under review. So this is radar. This is the UK Rare Renal Registry, and it's all millions of different well, not millions, less than 100, but quite a lot of rare diseases, all that are registered for data collection, really, and it's anonymous, so there's no risk to the people who subscribe. And these are the number of people registered with cystinosis over the years. There will be some patients on this that have died because this isn't censored for death, but it gets to about 180 odd at the latest count on this one. And then when people come to this hub clinic, we've got some pages where we enter data into radar. And so we talk about urine output and the gastrointestinal system, musculoskeletal, and the eye clinic and everything else. So this data then all goes into radar. And then in the long term, we'll have some really great follow-up data. It's broadly mapped to all Cervase's European database. So there should be an ability in the future to put it all together. And so, as I mentioned, we think hubs should be accessible to all patients. And we work together. We can always work together better. We're always very busy, but we can always work together a bit better. We refer in between the hubs, that's not a problem. We have shared clinical learning, quality control and improvement, and research that you've heard a lot about today, which is brilliant. And we can definitely try and move some of these things forward altogether in the three main hubs. And also, those of you that are not in the UK will probably, hopefully, be under a specialist hub in your country if you've got one. And we can work together with the hubs in Germany, France, Holland, Belgium, etc., to try and improve the care for everybody. This is the link to the website, but as I say, just put it into Google, you'll find it. Yeah, so that's my last slide. Thank you to everybody. Thank you. This is my the adult team, the pediatric team at the Evelina, my colleagues in the other hubs, international colleagues, pharmaceuticals, Cystinosis Foundation UK, CNE, and the conference organisers. And yeah, thank you for listening.
Thanks very much, David. The hard work has paid off. Can I just ask do a quick survey of the audience? Of those patients who live in England, are there any that have not been seen in a hub? Okay, so that's really helpful. And that's a very quick win. We can solve that one quite quickly. So come and talk to David or I, and we'll get that sorted out. Any questions for David? Yeah.
Hello. First of all, I just wanted to say, I think my experience with the hub, I attend your cystinosis hub, and I think it's a great concept. And as it's been developed, I think it's going to continue to improve the life of people with cystinosis in having a kind of one-stop shop for things. When you mentioned about dentistry and dermatology, I did wonder because I was contacting someone in America a few years ago, and I know some people with cystinosis get kind of, it seems like a skin, like almost like an acne scarring type look. And I spoke to him. He said that was to do with the cysteamine within the skin. Obviously, I know a lot of post transplant people see dermatologists in regards to sun protection, but I wondered that, and also with dentistry, I've heard that there can be issues with the dentistry due to some of the drugs that we take or some of the side effects of the condition. Is that something that could be looked at in future, including?
Thanks, Avril. I'll start with dentistry because it's so difficult. I mean, anybody trying to see a dentist in the UK knows how very difficult that is. I think we just felt that was a little bit of a bridge too far, and also we don't really have any sort of control over that. You see what I mean? We can kind of persuade, cajole, and bribe our colleagues pay our colleagues to come to the clinic, but we don't really have a facility to do that with the dentist, unfortunately. So. No, I definitely hear you, but in terms of the hub, I think that's pretty tricky. And even within my hospital, Guy's Hospital, which is one of the largest dental schools in the UK, just even getting into that can be really difficult for anybody. So I hear you, but it's a really big problem. Dermatology is an interesting one, isn't it? Because there are some cystinosis-specific issues, but I don't quite know how to make that work, because I think because what's the intervention you see? So if you've got this particular issue, what are you going to do about it, and who has that expertise? I'd be interested to hear from anybody else who knows the answer to that.
David, just to add to your point, and thank you, Avril. So I help to run the clinic at Birmingham Children's Hospital, and for dental and dermatology, although we can't get those specialties to come to the clinic, we do try and advocate for our patients that have those issues, and we try where we can to get them into a clinic pathway. We're really lucky that we don't have long wait times to get our patients into those services. We often do have barriers in ongoing input from those services, but that's a wider NHS issue that David was alluding to. And once we start to see patterns through a national format, like we do with other services like BBS, and as patients start to give us their feedback, that provides more leverage to ask other specialties to come and help us out, really. So keep doing it. It's really good to hear it.
Thank you very much for the presentation and great work. This is really, really incredible. I wish we had this where I was at in the States. I was curious, just thinking about, like, if you were to try to do something like this in the States, how difficult was it to recruit non-nephrologist specialists to come to the clinic? And did many of them already have expertise with cystinosis, or are many of them learning as they join these hubs?
Variable. So speaking for Graham, his unit already had quite a few people in your clinic, didn't you, Graham? Whereas for me, most of my recruits, some of whom you would have heard yesterday, were fairly new to cystinosis, but how were sort of academics with interest in similar conditions. So it's a fair point.
I think it raises a really important issue, which is that, as David said, we had some links, but it was fairly informal. And to make this really work, it has to be properly supported, funded, organized. Otherwise, it lasts for a while and then somebody retires and it all falls apart. So I think what David's done is make sure that this is set on a permanent footing.
I've got a couple of points, or one's a question and one's a dose of irony. So the first one's about scripts. You mentioned you can get orders for Cystagon and Cystadrops done from the hubs. I didn't really realize until Tina mentioned last night that in England, I kind of half knew, but you're paying for scripts in England. We don't do that in Scotland. So does that negate any cost element by doing that through the hub, just for even those particular parts that help patients?
I'm not sure. So I'm not, maybe, does anybody know about, does the home delivery service, do they still have to pay a prescription charge for the home delivery service? I'm not sure I even know the answer to that. I would imagine most patients who have got multiple prescriptions would have the, what's it called, the prepayment card, prepayment certificate, which would hopefully mitigate some of that cost because you'd be on lots of different medications. Yeah.
One to think about.
Thank you.
And the second point was about the situation we've got in Scotland. So as you say, we're served brilliantly by Glasgow and out in Edinburgh and Aberdeen and stuff. But as we're applying for PROCYSBI through the Scottish Medicine Consortium, we are considered a rare disease, lovely. We could be, or an orphan disease, we could be considered an ultra orphan, but there are 4 conditions to meet those requirements. And it's like number of patients. I can't really remember the other 2, but there is one that really stuck with me, which is you have to have a national designation centre. And I'm like, well, wait, we don't have that in Scotland because we are so rare that we wouldn't even think about setting up a national designation in Scotland, but we can't be considered ultra orphan without that. So I have written, complained, voiced, whatever, but I just don't know how to show them that that doesn't make any sense.
You've got a Scottish liver transplant centre, you've got a Scottish heart transplant centre, there's only one of them. So is it semantics that you can argue that you call the Glasgow centre the National Designation Centre and then you tick that box? I don't know what the bureaucratic requirement would be to rename it that, but it seems to me that you've got one, just got to call it that.
Yeah, it's a bugbear.
Well, we could probably take it offline and talk more about it, because the Department of Health, which does cover the whole of the UK, has one single rare disease strategy. And so I think it's definitely worth discussing. There is a way around. I think we've run out of time, actually. So I apologize, and we can talk over the break. What we've seen is some fantastically exciting hope for the future, for changes in treatment, improved care, improved treatment in many different areas. And then you've just heard from David how the organization of care can be delivered to improve outputs, including, I think, research as well through the centres, as David mentioned, because for the first time there is a mass of critical mass of people coming to a small number of areas where research can be focused in. So the clinical and the research will often end up together. So I'd just like to thank all of the speakers for a really fabulous afternoon, for your great contribution to everything, and a hopeful future ahead. Thank you.